Hello everyone. We've been looking at various cultures I've prepared for the past few weeks, and as you can see, it can be very hard to look at a microorganism in isolation. Even though aseptic procedure, when followed correctly, can prevent issues of contamination, a microbiologist may employ a medium that is either differential or selective toward a specific organism or organisms. Before we go into the different types of media we will be using today, let's first discuss what resident and transient flora are. Resident flora are, as you guessed it, residing in or on our bodies. Resident flora serve a purpose for each of us and are present on and in the various parts of our bodies. Transient flora are those organisms that we may pick up from our interactions with others and the environment that are not normal residents of our bodies, but that become part of our flora for a short period of time. Our skin is our largest organ and is our primary defense. Whenever we get a cut or abrasion, we are exposed to a number of microorganisms that try to take up residence in our bodies. Additionally, our resident flora can also cause disease, like in situations where our immune systems are compromised or when antibiotics have caused them harm. Now let's discuss the three types of mediums you will be working with today. The first medium you'll be using to conduct the hand washing experiment is the brain-heart infusion medium. This is a general purpose medium that is highly enriched. This medium will not inhibit any microorganisms and will provide for a suitable medium to allow you to detect the presence of microorganisms before and after hand washing. To conduct this procedure, divide your brain-heart plate into 6 sections as drawn out on the board. Your partner will turn on the water and handle the plate for you while you conduct each wash and place your finger on each of the sections of the plate. Use the sterile paper towels to dry your hands after each wash. Your partner will supply you with this. L1 and R2 will be for the left and right index fingers initially. L3 and R4 will be after you've washed your hands the first time, and L5 and R6 will be for the final wash. Be sure to use the DI water to wash your hands. After you're done, invert your plate and incubate. Let's now address what it means to be a differential and selective medium. Differential media allows the microbiologist to distinguish microorganisms from one another based on how they appear on the plate. Selective media allows us to select for a specific microorganism by only permitting for a certain one to grow. The first of these two mediums that you will be using today is mannitol salt agar. Mannitol salt agar contains a high concentration of salt, which makes this medium selective for staphylococci, but also contains mannitol that, when metabolized by pathogenic Staphylococcus aureus, produces a yellow color in the medium, differentiating it from non-pathogenic staph species. Today, you'll be dividing 2 plates in half and streaking a swab sample from your cell phone on one half of the plate and your nasal swab with the other half. Your partner will do the same to their plate. Your TAs will inoculate a plate with Staph epidermidis. After you're done, invert your plate and incubate. Tellurite glycine agar is the second medium that you will be testing. This medium is selective for pathogenic staph species and inhibits the growth of most other bacteria. It's differential by possessing potassium tellurite, which is reduced by pathogenic coagulase positive organisms to form black colony. Today you will be following the same procedure as with the mannitol salt agar. Your TAs will streak the Staph epidermidis on a separate plate. These are just a few of the mediums that can select for and differentiate microorganisms. Please take a minute to flip through our book of mediums to see what else is out there. I leave you with one final image. We all know about Pigpen from the Charlie Brown comics, but did you know that we are all basically a pigpen, no matter how clean we are? Our skin is constantly shedding off dead cells, and even though we cannot see it, we are constantly surrounded by a cloud of dead cells and microbes. Enjoy socializing now.